Somewhere inside the human genome, there is a genetic pattern that scientists cannot find anywhere else on the planet. Not in Nigeria, not in Ghana, not in Jamaica, not in Brazil, not in the United Kingdom. It shows up in exactly one group of people: Black Americans. And for a very long time, almost no one in the scientific world wanted to explain why. The technology to study it existed. The data existed. What was missing for decades was the willingness to say plainly what that data was pointing to. Because the explanation is not simply about biology. It is about history. It is about violence. And once you actually understand it, an ordinary ancestry test will never look the same to you again. Let's start with something uncomfortable. When scientists talk about the DNA of Black Americans, they're not describing one clean ethnic line the way you might describe Korean DNA or Norwegian DNA. They are describing something far more complicated. A genome that was not built slowly over tens of thousands of years in one region, the way most human populations were formed. Instead, this genome was built in a few hundred years, under extreme pressure, out of pieces pulled from many different places at once. To understand how unusual that is, you have to look at a number that is almost hard to picture. Historians believe that close to 12 million African people were forced onto ships during the transatlantic slave trade, a system that ran from roughly the 16th century all the way into the 19th. These were not people from one nation or one culture. They came from many different regions along the west and central African coast, from what is now Senegal, from the coast near Benin, from the Gold Coast, from Angola, from the Congo Basin. They spoke different languages. They followed different customs. They belonged to dozens of separate ethnic groups that, under normal circumstances, would never have combined into a single population. But normal circumstances did not apply here. They were forced together. Researchers who study mitochondrial DNA, the genetic material that passes only from mother to child, have traced maternal lineages back through thousands of individuals across more than 100 African population groups, trying to figure out exactly where these ancestral lines began. Think about that number for a second. Over 100 distinct African population groups all feeding, eventually, into what would become one shared American identity. That is not how ancestry usually forms. That is a forced merging, squeezed into roughly 10 to 15 generations under conditions that almost no other population in recorded history has had to survive. That compression left a genetic mark that is truly unique, but the African side of this story is only one part of it. Layered directly on top of that incredible internal diversity is a second thread, and this one is harder to talk about. Scientists who study genetic mixing, what researchers call admixture, have found that the average Black American genome includes somewhere between roughly 15 and 25% European ancestry, though the number varies a lot from person to person. Some individuals carry far more, some carry very little. That range tells part of the story. But the real story is in how that European ancestry got there in the first place. When researchers compare the Y chromosome, which passes from father to son, against mitochondrial DNA, which passes from mother to daughter, they find a pattern that cannot be explained by ordinary relationships or voluntary mixing between communities. The European genetic contribution came overwhelmingly through male lines. That tells us the mixing was not mutual. It reflects a one-directional pattern shaped by the power structure of slavery itself, where enslaved women had no legal power to refuse. Scientists have a clinical name for this pattern: sex-biased gene flow. But behind that clinical language sits a documented history of coercion, still visible today in millions of living genomes, more than a century and a half after slavery formally ended. The DNA itself holds a record that no historical document fully captured. There's a third layer to this story too, one that most people have never even heard of, and it kept reshaping the picture long after slavery ended. Between the early 1900s and the 1970s, roughly 6 million Black Americans left the rural South and moved to cities like Chicago, Detroit, New York, and Los Angeles, in what is now known as the Great Migration. Scientists studying genetic relatedness across the country have found that this movement did more than change where people lived. It actually changed the genetic makeup of the population itself, creating different ancestry patterns in different regions and connecting communities that had been separated for generations. In other words, this genome kept changing over time, not because of biology, but because of economics, survival, and the search for a better life. So when you put these three forces together, the forced mixing of dozens of separate African ethnic groups, the coerced and heavily male-biased European ancestry that came directly out of slavery, and the internal reshaping caused by mass migration within the United States, you end up with a population whose genetic makeup cannot be copied anywhere else in the world. Not in West Africa, where individual ethnic groups have stayed largely separate. Not in the Caribbean, where different colonial systems pulled from different parts of Africa in different ways. Not in Brazil, not in Britain. Every one of those populations has its own distinct mixing story. None of them match this one. This is not randomness. This is a specific, traceable, mathematically distinct genetic pattern that belongs to one population in human history. Now here is where the science becomes even more layered, because this is not only about where the ancestry came from, it is about what that ancestry is quietly doing inside people's bodies right now. Back in the 1990s and 2000s, medical researchers were trying to solve a stubborn mystery. Why does kidney failure affect Black Americans at close to 3 times the rate it affects white Americans? Digging into this question led scientists to a gene called APOL1, and what most people don't realize when they hear the phrase genetic risk is where that risk actually came from. The answer flips the whole story around. The APOL1 variant was never a flaw. It was originally a shield. Thousands of years ago in West and Central Africa, a version of this gene appeared that protected people against a deadly parasite responsible for African sleeping sickness, a disease that can kill without treatment. People who carried this protective version were more likely to survive infection. Over many generations, natural selection did exactly what it always does. It spread that protective gene through the population, because the people who carried it lived long enough to pass it on. This was evolution solving a real life-threatening problem in the environment where it was needed. But then everything changed. That same protective gene traveled across the Atlantic through the same forced migration that shaped the rest of this genetic story, and it landed in a completely different environment. One without sleeping sickness, but with entirely different health pressures. In this new setting, the very same gene variant that once saved lives from a parasite now raises the risk of kidney disease. Today, researchers estimate that roughly half of Black Americans carry at least one copy of this risk variant, and about 13% carry 2 copies. Carrying 2 copies significantly increases the risk of certain kidney conditions, including a disease called focal segmental glomerulosclerosis, as well as kidney failure linked to high blood pressure. Think about what that means. A gene that once meant the difference between life and death from one disease now means something completely different centuries later on a different continent, inside the bodies of people whose ancestors never chose to make that journey. This is where the story stops being only about ancestry and starts being about medicine, and the implications are huge. This gene variant is found almost only in people with recent African ancestry. A genetic technique called admixture mapping, the same kind of method used to trace ancestry, has become one of the most powerful tools scientists have for hunting down disease-related genes in mixed ancestry populations like Black Americans. Because of their unique genetic structure, Black American genomes have turned out to be scientifically extremely valuable for identifying disease risk factors that would be much harder to find in more genetically uniform populations. The very history that created this one-of-a-kind genome is now, generations later, helping researchers understand and try to treat diseases that disproportionately affect the descendants of that same history. But before this starts to sound like a simple success story, there is a harder truth underneath it, one that researchers themselves have been raising more and more urgently. According to population geneticists and public health experts who study this exact issue, the scientific narrative around genes like APOL1 has repeatedly been twisted into something dangerous, the false idea that Black Americans are somehow biologically destined for worse health outcomes, as if genetics alone explains the disparity. Many medical researchers have pushed back strongly against this idea, pointing out that the frequency of this gene variant varies enormously across different Black communities and should never be treated as some kind of single racial marker. The real picture is far more complicated and far more uncomfortable for anyone looking for a simple genetic explanation. Genetic risk is only one piece of a much bigger puzzle. Access to early screening, quality of health insurance, environmental exposure, chronic stress, and decades of documented gaps in medical treatment all stack on top of any inherited biological risk. Some researchers argue that focusing too much on the genetic explanation for kidney disease disparities risks becoming a convenient distraction from the deeper structural and systemic causes that have nothing to do with DNA at all. Think about what that means. The very same data that reveals something scientifically remarkable about Black American ancestry can, if handled carelessly, be twisted into a story that lets much larger systemic failures off the hook. The science itself is neutral. What people choose to do with it is not. And this tension between a genuinely extraordinary scientific discovery and the risk of that discovery being misused is exactly why so many researchers have been careful about how they talk about it publicly. Now consider what happens when this unique genetic architecture meets an industry that millions of people trust without question, commercial DNA testing. These companies sell customers a clean, confident-looking pie chart, claiming to show exactly which countries their ancestors came from. But here is where things get messy again. Black American ancestry does not map neatly onto modern African borders the way these companies often imply. The ethnic groups that contributed to this genome were spread across regions that did not match today's national borders, even at the time of the slave trade. And many of those original populations have themselves changed, migrated, or mixed together over the last 2 centuries. What most people never realize when they open an ancestry report is that those confident-looking percentages are statistical estimates, built using reference populations that are themselves imperfect stand-ins for real history. The DNA itself is real. The neat story built around it is often a simplification being sold as certainty. This is also where a real shift in power shows up in this story. Not political power, but the authority to explain a population's own history back to itself. For most of the 20th century, the major institutions studying African American genetics were overwhelmingly led by researchers working with reference data sets that were themselves incomplete, often built from limited sampling across Africa that failed to capture the continent's true genetic depth. Researchers now working to correct this imbalance point out that Africa actually holds more human genetic diversity than any other continent on Earth, simply because humanity originated there and has the deepest evolutionary history there. And yet, for decades, African genomes have been among the least studied in global genetic databases. That means for a long time, scientists were trying to explain the most complex mixed ancestry genome in the Western Hemisphere, using a reference map that was itself badly incomplete. The very tool used to interpret this unique genetic story was, for most of its history, never built to properly understand it. That imbalance is finally starting to change, but slowly, and the effects of decades of underinvestment are still being felt today. Newer, large-scale genetic databases have started expanding their African reference data, refining ancestry tracing methods, and building more accurate models of exactly how African, European, and in some cases Native American ancestry combine across different Black American individuals and regions. This matters enormously for medicine, because genetic risk prediction tools, the algorithms increasingly used in hospitals to estimate a patient's likelihood of developing certain diseases, have historically performed worse for people of African ancestry, precisely because they were built on data that underrepresented this kind of complex mixed ancestry. In other words, the same historical exclusion that shaped this genome in the first place has, generations later, quietly shaped how well modern medicine can even read that genome accurately. So here is where all of these threads start to connect, layer by layer. A genome forged from the forced merging of dozens of distinct African ethnic groups, a pattern of European ancestry carrying the genetic fingerprint of coercion, a migration-driven reshaping of ancestry across generations, an ancient protective gene turned into a modern health risk by a change in environment nobody chose, a scientific establishment only now catching up to the complexity of what it is studying, and an entire commercial industry selling simplified certainty about a genetic story that resists simplification at every step. This is not one single discovery. It is a chain of discoveries, and each link reveals something new about how history, biology, and power intersect inside a single strand of DNA. But here's a part of this story that almost nobody talks about, and it might be the most important twist of all. This unique genetic structure does not just record the past. It is actively shaping the future of medicine for everyone, not only for Black Americans. Populations with complex mixed ancestry like this one are uniquely valuable in genetic research for a very specific technical reason. When ancestral chromosome segments recombine across generations of mixing, they create natural experiments that help scientists pinpoint exactly where in the genome disease-related variants are located. Admixture mapping, the same technique that helped researchers identify the link between APOL1 and kidney disease, depends entirely on studying populations with this kind of layered ancestry. That means research built on Black American genomes has already contributed, and will likely keep contributing, to broader breakthroughs in understanding the genetic basis of disease across all populations. A population born out of history's cruelty is now quietly powering some of modern medicine's most important discoveries. And yet, even with all of this scientific importance, representation remains a serious, well-documented problem. Multiple studies looking at genetic research on Black populations have pointed to ongoing underrepresentation in clinical trials, in genomic databases, and in the reference panels used to build diagnostic tools. Think about what that actually means. The population whose genetic complexity makes it scientifically invaluable is, at the same time, the population most likely to be left out of the very studies that could benefit from that complexity. That contradiction sits right at the center of this entire story. Extraordinary scientific value and ongoing institutional neglect, existing side by side, generation after generation, with neither one canceling out the other. Now, step back for a moment, because there's a bigger, quieter point underneath all of this worth sitting with. Every population on Earth carries a genetic record of what happened to its ancestors. Most of those records were written slowly, over tens of thousands of years, through ordinary migration, ordinary intermarriage, and gradual adaptation to climate, food, and disease. This one was written differently. It was written in under 400 years. through forced transport across an ocean, through the merging of dozens of cultures that never chose to merge, through a documented pattern of coercion embedded in the male lineage, through a migration born from the search for safety and opportunity after slavery ended, and through a modern medical system still trying to catch up to the complexity it inherited. That is not an ordinary genetic history. That is a genetic history compressed under extraordinary hardship. And its uniqueness is not something to treat as exceptional in a celebratory sense. It is a scientific record of exactly what was survived. This is exactly why how we talk about it matters so much, because the same discovery can be told in two very different ways. Tell it one way, and it becomes a story about biological difference used to separate one group of people from another, one of the oldest and most dangerous patterns in the history of genetics, a pattern that has been used before to justify segregation, forced sterilization, and pseudo-scientific racial hierarchies that legitimate science spent most of the 20th century trying to dismantle. Tell it the other way, and it becomes something much closer to the truth. A story about how history itself gets written into biology, how survival leaves a signature, and how a population forged through catastrophe produced a genetic architecture that is now helping unlock medical mysteries for millions of people who will never know the names of the ancestors who made it possible. What most people miss when headlines shrink this down to "scientists discover unique black DNA" is that the real discovery was never really about difference for its own sake. It was about specificity, a precise, traceable, historically explainable genetic signature built from documented events, not some mysterious racial essence. Every part of it has a cause. The African diversity has a cause, the European ancestry has a cause, and an uncomfortable one. The regional variation has a cause. The Apon risk has a cause. None of it is random, none of it is destiny. All of it is history, written in a language most of us only recently learned how to read. So, where does that leave things today? In a research field that is still catching up to its own blind spots. Reference databases are growing, but slowly. Ancestry models are getting more accurate, but they are still built on foundations that took far too long to include proper African sampling. Clinical trials are becoming more diverse, but representation gaps that took generations to create will not close in just a few research cycles. And commercial ancestry companies keep selling tidy percentages to millions of customers who have no idea how much historical violence and statistical guesswork sits behind every number on that report. The science is moving forward quickly. The reckoning over what to actually do with it is moving much more slowly. And there is a question underneath all of this that researchers themselves have not fully answered yet. If this genetic architecture really is one of a kind, shaped by a specific historical catastrophe that can never be repeated or undone, what else is waiting to be found as reference databases finally catch up? What other disease risks, protective genes, or evolutionary trade-offs are still sitting undiscovered inside a genome this complex, waiting for tools sophisticated enough to finally read them? And perhaps even more urgently, as genetic risk scores become a normal part of everyday medicine, who gets to decide how this information is used? To finally close health gaps that have lasted for generations, or, if history repeats itself, to quietly reopen a door that science spent the last century trying to shut for good. Nobody currently studying this genome claims to have all the answers. The data keeps growing. The models keep improving. And every year, researchers uncover another layer of a genetic story that was, quite literally, written by force, carried across an ocean, reshaped by migration, and is now sitting inside the cells of tens of millions of people who never asked to inherit any of it, but whose bodies, whether they realize it or not, are still holding on to every part of how they got here.